Cancer, a formidable foe, has challenged medical science for decades. Our understanding of cancer has evolved, and we've learned that it's not just about abnormal cells growing uncontrollably. It's also about the complex interplay between cancer cells and our body's natural defenses, the immune system. For years, treatments like chemotherapy and radiation were the mainstays, often with debilitating side effects. Then came immunotherapy, a revolutionary approach that harnesses the power of our own immune system to fight cancer. Immunotherapy doesn't directly attack cancer cells like traditional therapies. Instead, it empowers our immune cells to recognize and eliminate cancer cells, offering a targeted approach with potentially fewer side effects. This field has witnessed remarkable advancements, and among the most promising innovations are bispecific and trispecific antibodies, engineered proteins designed to enhance the precision and effectiveness of cancer immunotherapy. Our immune system is a complex network of cells and proteins that defend our bodies against invaders like bacteria and viruses. One of the key players in this defense system is the antibody, a specialized protein that can identify and neutralize specific targets known as antigens. Antibodies are like guided missiles targeting specific enemies. In the context of cancer, however, our immune system sometimes fails to recognize cancer cells as threats. This is where bispecific antibodies come into play. These engineered antibodies are designed with two binding sites, each capable of recognizing a different target. Imagine a bridge connecting two different entities. That's essentially what a bispecific antibody does. It can simultaneously bind to a specific antigen on a cancer cell and a receptor on an immune cell, effectively bringing them into close proximity. Let's understand this with an example. Blinatumomab, a bispecific antibody approved for treating a type of leukemia. In this case, the bispecific antibody acts as a bridge between two key players, CD19 antigen. This antigen is found on the surface of leukemia cells. CD3 receptor. This receptor is present on T cells, the warriors of our immune system responsible for directly killing infected or cancerous cells. Blinatumomab binds to CD19 on the leukemia cell and CD3 on the T cell, pulling them together. This close encounter activates the T cell, unleashing its cytotoxic potential to destroy the leukemia cell. Section 4. Trispecific antibodies, taking precision to the next level. Building upon the success of bispecific antibodies, scientists have developed trispecific antibodies, engineered proteins with three binding sites. This allows for even greater precision in targeting cancer cells while minimizing effects on healthy cells. These antibodies can simultaneously engage multiple targets, leading to a more potent and controlled immune response. Section 5. Describe how trispecific antibodies work, including an example. Imagine a trispecific antibody designed to target a solid tumor. First, this antibody could bind to a specific antigen on the tumor cell. Second, it could also engage a receptor on a T cell, activating the T cell as we saw with bispecific antibodies. Additionally, this trispecific antibody could be engineered to block a protein that the tumor uses to evade the immune system, further enhancing the anti-tumor response. This multifaceted approach highlights the potential of trispecific antibodies to overcome the complexities of cancer. Section 6. Unlocking the potential benefits of bispecific and trispecific antibodies. Bispecific and trispecific antibodies offer several advantages over traditional cancer therapies. Enhanced specificity. These antibodies can precisely target cancer cells while sparing healthy cells, minimizing side effects. Increased immune cell activation. By bringing immune cells in close proximity to cancer cells, these antibodies enhance the immune system's ability to recognize and destroy cancer cells. Overcoming immune suppression. Some cancers can suppress immune responses. Bispecific and trispecific antibodies can be designed to counteract these suppressive mechanisms, boosting the effectiveness of immunotherapy. While still an area of active research, early clinical trials have shown promising results in treating various cancers, including leukemia, lymphoma, and multiple myeloma.